All right, today we are going to finish the digestive system. We've gotten all the way down, the food has gone all the way through the stomach. Just a real quick review. When it's in your mouth, it's mixed with what juice? Saliva. What enzyme is found in saliva? Salivary amylase. What food group does it break down? Carbohydrates. Specifically, it breaks down starches into disaccharides. There's also a mucus component to saliva, and so that lubricates the food, and now it's this lubricated mucousy mass of food that you swallow. What is that called? A bolus, and it moves through the pharynx and esophagus through what sphincter into the stomach? What's the name of the sphincter at the top of the stomach? The cardiac sphincter, or you can say lower esophageal sphincter. Now it moves into the stomach and mixes with what juice? Gastric juice that has what big enzyme in it? Pepsin, thank you. And pepsin has to be activated first. And what activates it? Good, hydrochloric acid, produced by what cells of the gastric glands? Parietal cells. What's the name of the cells that produce the enzymes in gastric glands? Not parietal cells, but chief cells. Okay? See how detailed this can be? What's the name of the folds in the mucosa of the stomach? The rugae. Rugae. Okay? As the gastric juice mixes with the food, now it becomes this pasty, semi-solid, liquidy stuff that moves into the small intestine. What is it referred to now? Not a bolus, but chyme. And as it moves into the small intestine, what sphincter does it have to pass through? The pyloric sphincter. Yes. When it gets into the small intestine, one of the juices it encounters is pancreatic juice. Name some of the components of pancreatic juice. Ooh, you need to know that. Go study that. It'll be on your test. Pancreatic amylase is one. Trypsin is one. Chymotrypsin, carboxypeptidase, those last three. Trypsin, chymotrypsin, carboxypeptidase work on what food group? Proteins. Therefore, because they work on proteins, they're aggressive. They have to be released in an inactive form. What is the chemical that activates the inactive form of trypsin? It's called enterokinase. It's actually produced at the small intestine. So trypsin doesn't get activated till it gets to the small intestine. And then once it's activated, that in turn, its presence activates the other two protein digesting ones, chymotrypsin and carboxypeptidase. There's another enzyme called pancreatic lipase. What does it work on? Fats. So does it work on the fats in the pancreas? Think about this before you blurt out an answer. Does pancreatic lipase work on fats in the pancreas? No. It's got to get to the small intestine, so it's working at the small intestine. Okay, another organ that produces a juice that empties into the small intestine is the liver, and that's where we're going to start today. If you looked in the body cavity, in the ventral cavity, just under the diaphragm, the liver takes up the bulk of that upper part of the abdominal cavity, right below the diaphragm. It has two main lobes and then a couple of smaller ones. You're not going to have to name any of the lobes. But internally, each of the lobes is made up of little microscopic units called lobules, hepatic lobules. If you ever see the word hepatic, that means liver. Okay, hepatic means liver. Okay, hepatitis is a viral infection of the liver. Okay, but anyway, the microscopic lobules in the liver, that's the functional unit of the liver, and that means that's the smallest part of the liver that does everything a liver does. Okay. Now, when you look in your book, if you were to look on page 673, I don't believe I have that table. Nope, I do not. So it'd be nice if you looked at page 673. There you can see the big functions of the liver on table 17.7. And if you don't have your book out, a lot of you do, I see. That's great. I'm going to read some of these to you. One of the functions is carbohydrate metabolism. So it says it polymerizes glucose to glycogen. That means it converts glucose to glycogen. It breaks down glycogen to glucose. It converts non-carbohydrates to glucose. So in other words, it builds and it breaks down carbohydrates. It does the same thing to lipids. It builds and breaks down lipids. It builds and breaks down proteins. It stores some chemicals. It filters your blood of foreign things. It removes toxins from your blood. And it produces bile. All right, and so I've said a lot of that right here. Okay, the functions of the liver. The reason the liver can do all of these things is because blood coming from your digestive tract makes its way to the liver. So that nutrient-rich blood that is loaded with these absorbed carbohydrates and lipids and proteins, that blood makes its way to the liver as it leaves the absorbing organs of the digestive tract, like the stomach, the small and the large intestine. All that moves into the liver. We talked about that in lab when I mentioned to you that hepatic portal circuit. In lab, we were naming the blood vessels. All right, and so this nutrient-rich blood that's deoxygenated, but it's still nutrient-rich from your digestive tract, runs through the liver before it gets to the vena cava, the inferior vena cava. And at the liver, a lot of those nutrients are pulled out of the blood and processed. Okay, so that's why your liver is one of the main places in the body where you store a lot of glycogen. The other is in your skeletal muscles. Okay, and so that by itself, that first line, carbohydrate, lipid, and protein metabolism, that's a huge, huge, huge function of the liver. Okay, but it also does a lot of other things for you. It stores because it's getting all these nutrients from the digestive tract. That's a good place to store some excess. So it can store glucose in the form of glycogen. It can store some vitamins and some minerals like iron is stored there. And then because it's blood moving through, this is a great place for some phagocytes to reside. So in the liver, there are a lot of phagocytes that can eat up bad things in your blood. So it helps to filter your blood of some of the damaging substances. If there are chemicals in your blood that are harmful to you, they can be removed at the liver. So that's the detoxification. You hear about long-term alcoholism resulting in a condition called cirrhosis of the liver. All right, what that is, alcohol chemically is a lot like a simple sugar. It's absorbed quickly, even can be absorbed at the stomach. And the first place it's going to go is the liver. And so if the liver, this is the way it's been described to me, if the liver has a choice between alcohol and glucose to break down, it's going to choose the alcohol. Evidently, it's easier. And so what happens to that glucose, as much of it as possible gets stored as glycogen, but there's a limit to the glycogen storage in the liver. So any excess glucose is going to be stored as fat. And that fat buildup over time, initially it's called a fatty liver, but over time it can lead to this condition called cirrhosis of the liver. Someone who has cirrhosis of the liver or a fatty liver, that doesn't mean that they're alcoholics, but that's one of the things that can happen if a person struggles with alcoholism. Okay. Other drugs can be removed at the liver. Okay. And then the last function I've listed, the secretion of bile, I started not because it's more important than any of these others, but that's this is the digestive function. See, the liver does a lot of things besides produce a fluid for digestive tract. Okay, the fluid that's produced by bile, the juice produced by the liver, is called bile, and bile contains a lot of the waste products of the activities that are listed there, but it also contains a chemical that aids in digestion. This bile is emptied into the small intestine along with pancreatic juice. So we're going to look at the chemicals. We're going to name them in a minute. They're easy. But what they do to your digestive system is it helps your small intestine emulsify fat. Now, you may not know what that word emulsification means, but let's say if you have just eaten a meal that's high in fat, right? That fat, lipids don't dissolve in water, so you get this little blob of fat. Let's say that fat right there is in your small intestine. It hadn't been broken down yet, has it? Okay. But so the lipases, like pancreatic lipase, they can only get to it at its edge, right? Okay. So do you see how it would take a long time for it to break it all the way down to the middle? Okay. But if that big fat droplet is broken down into a bunch of little tiny ones, do you see how this increases the surface area that the lipase could work on? So lipase can break down a bunch of little tiny drops of fat more efficiently than it can break down one big old lump of fat. And so taking it from this to this, that's called emulsification. And bile aid helps in that process. Okay. It's like chewing the fat. Yeah. it's like your teeth do. They break down big pieces of food into smaller pieces. It's a type of mechanical digestion. It's not chemical, it's mechanical. You temporarily emulsify fats if you mix oil and water, the oil goes to the top. If you shake it up, it's temporarily dispersed in little droplets. That's a type of emulsification, but this is more of a microscopic emulsification. It still doesn't change it into fatty acids and glycerol. It's still full-fledged triglycerides. But it's in a form, it's in little pieces so that the lipase enzymes that are going to chemically break it down can work more effectively. Okay, so here's your liver. You don't have to name the ligaments associated with the liver. The little green thing that you see underneath the right lobe, that's the gallbladder. That'll be the next organ we talk about. If we looked at it through the microscope, this is what you would see. So this is a lobule, this is a lobule, this is a portion of another lobule, and then you see a bunch of them drawn right here. Do you see in the middle of each lobule you have this opening right there? Right there, here you can see it. All right, that's called the central vein. All right, and so that's where some blood's going to flow. Okay, so watch. The hardest part about the liver is to figure out the blood flow through it. Okay, so imagine it macroscopically. Now this is from your lab information. Remember, you have your aorta has branches such as the superior and inferior mesenteric arteries, and they branch to the digestive organs through the mesentery to supply them with oxygen and nutrients, and then picks up CO2, but it also is picking up the absorbed nutrients from the food you're eating. Okay, and then that blood makes its way into the superior and inferior mesenteric veins, also a gastric vein from the stomach, which you didn't name in lab. But all of those veins coming from the digestive tract pool together at one called the hepatic portal vein that goes to the liver. And what you're seeing on this picture right here is a branch of that hepatic portal vein. It branches to feed all of these lobules. Okay? And so as this blood makes its way into the lobules, see, it's in these little, they call them little sinusoids, which is not a big deal on the test, but it feeds through these blood vessels and empties into the one in the middle, which is the same thing as that one right there. So here's a branch of the hepatic portal. It feeds through here to get into the central vein. And as it does, nutrients that are in it, like glucose and amino acids, they are taken up by these hepatic cells, okay, and processed with all those metabolism things, okay. There's also macrophages lining those sinusoids, and so by the time it makes its way to the central vein, it's more filtered, okay. And then that collects with other central veins to ultimately lead to a hepatic vein that empties into the vena cava, okay. All right, you also can see blood from an artery. This is oxygenated blood that mixes with the deoxygenated blood from the digestive tract. So, it makes its way to the vena cava also through that same way. This is a pretty interesting flow through the liver, in my opinion. And then, do you see the not the green that I drew, but the green that the artist that did this picture? Do you see these little green areas, like right here? And these are tiny little bile ducts. So look at all these little bile ducts. Uh, Bile is produced by these cells that gets into these little bile ducts that are going to leave the liver and ultimately go to your small intestine. Okay? So do you see how blood flows in one direction, the bile flows in the other? Okay. Right. Okay, here's another view that's a little closer. So you can see the blood flowing here and here, and it comes together. Oh, there's one of those little macrophages. It's called a Kupffer cell. That's one of the resident macrophages. And so it goes into the central vein. And then the bile moves into these little bile ducts to lead out of the liver. Okay, and then there's another picture. You can see the little Kupffer cells. I kind of went crazy on these pictures. I kind of like the liver. It's just hard to visualize. What you're looking at is microscopic right here. Okay. All right, and so what I've told you, that blood flow, it's all laid out here for you. Okay, so it's right there. But if you can talk through it on the picture, that'll help you, like always. And the little I think it's pronounced Kupffer cells. I think that's supposed to have 2 dots on it. I don't know. But anyway, those are the phagocytes. Those are some of those resident phagocytes, like you have some in the lungs. You have them in a lot of places. Okay, so what is bile? Okay, bile has a lot of things in it. Mostly it's just waste products from all those metabolic activities. Do you also recall that back when we did the blood chapter, that the liver is one of the main places in the body that breaks down old red blood cells? That destruction occurred at the liver, and from the red blood cells the hemoglobin was pulled out, and from the hemoglobin the heme unit was pulled out and was converted to biliverdin and bilirubin. Okay, biliverdin and bilirubin are some components of bile, and that's what gives bile its greenish yellow color. And those are the byproducts of hemoglobin breakdown. So that's what when it says bile pigments, that's that bilirubin and biliverdin. Of course, there's water in it. There's various electrolytes that you may have had in excess. Cholesterol. The liver is a wonderful organ to pull excess cholesterol from your blood. If you happen to eat liver, you're getting a lot of cholesterol. It's stored in the liver too. But it can be removed. So it can be a component of bile. But the digestive component is this one. And when I mean digestive component, I'm saying this is the part that affects digestive activity in your small intestine. So these are the bile salts. That's what emulsify the fats. They're just various, ions that have come together to form salts. But their presence on a fat droplet emulsifies it, okay, breaks it up into smaller pieces. Okay, so that's the digestive component. Make sure that you know there are no digestive enzymes in bile. It doesn't, all of the other juices that we talk about in the digestive tract contain some enzymes, but this one does not. Okay. All right, and then what is cholecystokinin? Is it an enzyme? No, it's a hormone. And do you remember it had a role in stomach activity? It tended to slow down things at the stomach, and it caused the pancreas to produce the enzymes, to release the enzymes in the pancreatic juice, like lipase. Okay, and what it does at the liver is help increase the release of bile. Remember, one of the triggers for cholecystokinin is the presence of fats in the small intestine. and doesn't bile emulsify fats. Okay. Okay. All right. And then the gallbladder is associated with the liver structurally and functionally. It's attached to the underside of the main lobe of the liver. It does not make anything for you, though. It just temporarily stores some of the bile, and while the bile sits there, water is absorbed from it. So it concentrates the bile to help it be a little more effective, and then periodically releases that. And its release also is due to this cholecystokinin. Okay. All right. So if we look at this picture on your left, here you're seeing the front of the liver, and there's a little piece of the gallbladder. If you flip that liver up to look at the back side of it, so here's the gallbladder, and I want you to notice this duct right there. Okay, that's coming out of the gallbladder. That's called the cystic duct. Okay. And then I want you to notice, this is a little complicated, or it's really not complicated, that duct and that one. Those are the ducts that are coming out of the two main lobes of the liver, and they're called hepatic ducts. See right there, hepatic ducts? And where they join together, that happens to be called, where they join together is the common hepatic. But do you see how then it connects to the cystic? And where the common hepatic and the cystic duct join together, that is the common bile duct. I've got all this in your notes. And the common bile duct is the one that empties into the small intestine. Okay? But it's where the cystic duct and all the ducts from the liver come together. And it's a long one. It's the big one. So if we see, we get you another view. This is the best view because this shows its association with the pancreas. So here's a lobe of the liver. There's a lobe of the liver. I knew I drew those terribly. But there's a, watch, hepatic duct. Where they come together is common hepatic duct. You can see it labeled right there, common hepatic duct. And then it connects with the cystic duct coming out of the gallbladder to form the really long one called the common bile duct. Okay? And so, do you see how the common bile duct empties through the same place the pancreatic duct empties through that sphincter of Oddi, or hepatopancreatic sphincter? Okay. So what happens is, as the bile trickles down from the liver, it can make its way up into the gallbladder, or it can go all the way through. If this is closed up, it'll back up and it can go into the gallbladder and can be stored there. Okay. You can live without a gallbladder because it doesn't make anything for you. So the liver will compensate for that and just, adjust the release of the bile, and it'll still just go through into the small intestine. Okay. You hear about gallstones sometimes. Probably most people have some little tiny gallstones, but all a gallstone is when the bile, some of the dissolved substances in it come out of solution, and they form little solid pieces. And as long as they're sitting in there, they don't hurt you a bit. But if they get big and happen to get lodged right there, it can be very damaging and can set up inflammation. And so how they treat that is either zap them with lasers, or go in and crush them, or just remove the gallbladder if it's really bad. Sometimes people have gallbladder attacks and then it goes away, and then several months later they may have another attack and then it goes away, and then several months later they may have another attack. What's going on is a gallstone may lodge, they have an attack, and then it drops back down into the body of the gallbladder. As long as it's in there, it's not hurt. You don't have any symptoms unless you get a whole bunch of them. Here's an x-ray that shows a whole lot of gallstones in someone's gallbladder. So. No, that one's probably right before its removal. Yeah. That's probably a diagnostic x-ray. Say, oh yeah, you need your gallbladder removed. Yeah. All right. So liver's a big deal, and the gallbladder. But, okay. Next organ. Now we're going to move back to the alimentary canal and look at the small intestine. Um, the food stays longer in the small intestine than it does the other organs. Could be several hours, um, because this is where most of the chemical breakdown and also most of the nutrient absorption occurs. So it's got to stay in there long enough for those processes. Your small intestine is smaller in diameter than the large intestine, but it's much longer. If all the muscles in it were relaxed, and if you could pull it out, there's a mesentery that holds it in there, but if you could take it off the mesentery, pull it out, do you know how long it would be? It's about 20 feet long. Very long. So, but anyway, here are the regions: the duodenum, the jejunum, and the ileum. Those are the three regions of the small intestine. The duodenum is the shortest part, and that's the one that connects to the stomach. Uh, and the and it's fixed. The jejunum and the ileum are the ones that are held in place by the mesentery, which you can see here. So here you see the stomach. So this is the duodenum. And then here's that mesentery. So the arteries in there, those are branches of the mesenteric arteries. And see how it holds the rest of this. And you've got jejunum. And I don't really know where the dividing point is to the ileum, but the ileum is the last part. And so the ileum connects to, then here's the large intestine, which will be the next organ we look at. Okay. All right. Now, at the top, you're seeing just a short little section of a small intestine, and you see it opened up. And one of the most noticeable things about it is that there's a lot of folds in its wall. So at first glance, you think, oh, that's rugae. But these are fixed, whereas in rugae, they can stretch out and then refold a little different conformation. Here, they're always fixed. And those fixed folds are called plicae circulares. And just that by itself completely increases the surface area of the mucosa, doesn't it? Do you see that? Increasing the surface area so that more things can be absorbed at one time. Okay. And then if you look at the magnified view, on the surface of each plica or each fold, you see additional little finger-like projections. And they are what the small intestine is famous for. They're called the villi. Singularly, that would be a villus. And you can see it's not just a total fold, it's just a little finger-like projection. If you were to actually see a real small intestine and open it up and look at the mucosa, it would look like velvet. You know, velvet has those little threads that stick up, like on a rug. All right, that's exactly what it looks like. The villi are macroscopic. They're tiny, but they're macroscopic. So do you see how that further tremendously increases the surface area? Do you see that? Okay. So without the plicae and without the villi, the small intestine would have to be a whole lot longer than the 20 feet, thousands of times longer than the 20 feet. Okay. Then, stay on this page. It's talking about the villi. There you can see some villi. Then this is showing you one villus. And do you see how the surface of it is simple columnar epithelium? Isn't that the absorbing epithelium? Okay, just like in the stomach, you had simple columnar. I see a few little goblet cells in there. Inside each villus, do you see that green structure that's labeled the lacteal? All right, that's a lymphatic vessel. And it's surrounded by blood capillaries. And so when nutrient absorption occurs, it occurs through the columnar cells, and then the nutrients either get into the blood or they get into the lacteal. We're going to see in a moment that the fats absorb into the lacteal. But remember that lymphatic vessels ultimately drain into the bloodstream, so it makes its way to the blood. Okay, but everything else, proteins, carbohydrates, electrolytes, those absorb directly into the bloodstream. Okay, on the right you can see an actual micrograph, and you can see the villi. They're not nice and neat. You can see them sectioned in bits and pieces. In between the villi, look at this. In between the villi, you have intestinal glands. In your notes on the page or two before this, there's another name for those. They're called crypts of Lieberkühn. Do you see that? Crypts of Lieberkühn? Okay. So the intestinal glands are to the small intestine what the gastric glands are to the stomach. They produce a juice. It's called intestinal juice. At least that's easy. And then there's mucus glands down in the submucosa that are not shown here. We'll see them in lab. They're called Brunner's glands, and they empty mucus into that intestinal gland. You actually can see some of those right in here. Some of those Brunner's glands right there. Okay. And then if we took one of these cells right there, just one of those cells, it's a columnar cell. This is what it looks like. And look at its free surface. It has microvilli, which is further increasing the surface area. This is extremely significant. The small intestine's surface area, I have no idea how big it is. I'm sure somebody's measured it somewhere. It's like many football fields, probably, of surface area for one person, or a football field, because of the plicae circularis, the villi, and the microvilli. And that's significant because so much nutrient absorption occurs. Okay. So now we're going to look at the intestinal juice and its components. Of course, there's always a water component, but there's mucus from the goblet cells, and then the Brunner's glands. Those are mucus glands in the submucosa that empty into the little intestinal glands. Okay, and then there's an enzymatic component, and so here's some more enzymes, but these are easy. Peptidases, it's obvious that does something with peptide bonds, probably, doesn't it? So it breaks proteins down into amino acids. Sucrase, maltase, lactase break down sucrose, maltose, lactose, which are disaccharides, and breaks those down into monosaccharides. Lipases, there's an intestinal lipase, just like there's pancreatic lipase, breaks fat into fatty acids and glycerol. So if you look at this, this, and this, do you see how these are breaking those macronutrients into small enough pieces now so they can be absorbed? So this is the finalization of the chemical breakdown. All right, and then enterokinase, we already named. That's the chemical that activates trypsinogen that's coming in with pancreatic juice. Okay. And then there's hormones. Oh, look, there's cholecystokinin again. But these hormones, this is just sort of a repeat of what you've already learned about hormones. It's just a little review. Gastrin is not on here. I should, because these are some hormones that are secreted by the small intestine, and gastrin is actually produced at the stomach. But if you wanted to add a fourth hormone, because you're learning about 4 hormones, you could add gastrin to this. Just know it's not made at the small intestine, it's made at the stomach. And it increases stomach activity. But somatostatin, we already talked about, that slows down secretion at the stomach. Cholecystokinin does a whole lot of things, doesn't it? It slows down stomach activity. It causes the pancreas to produce enzymatic pancreatic juice. It causes bile release at the gallbladder. Okay, and then secretin causes the pancreas to produce the bicarbonate ions. What's the purpose of bicarbonate ions in the digestive tract? I thought y'all studied. Raises the pH. It raises the pH. Okay. All right, so I mentioned to you about this absorption through the villi. Your book goes into more depth with it than your test will. Basically, all I want you to know is that everything is absorbed into the blood except for the fats. They're absorbed into the lacteal, okay? All right, so stay here. Here you see, these are the microvilli on a columnar cell, so everything's absorbed through here. Okay, and here's a lymph vessel, one of those lacteals. Okay, so your fatty acids and glycerol that are absorbed come together along with some proteins to form a lipoprotein called a chylomicron. We've never used that word, but a chylomicron, and that's the form the fat is in when it gets to a lacteal. It's in the form of chylomicrons. Those are types of lipoproteins. It says it on the page before this. Okay, and then everything else is going to be absorbed into the bloodstream directly. Okay. All right. Now, the greater omentum is interesting. We're done with the small intestine, with the insides of it. But if you look at this picture, if you were opening up somebody's ventral cavity and you remove the body wall, remove a little bit of the musculature that's there, then as you get below that, you really can't see the intestines because there's this fatty membranous stuff that covers it that's hanging down. It's almost like an apron hanging down from the stomach. It's called the greater omentum. And a lot of times on pictures that's been removed so that you can see what's underneath it. But that provides some physical and protection for the abdominal organs. But it's kind of interesting. It's just sort of a side note to all this. There you see it. All right, now here's a table that has all these enzymes we've talked about. And it's overwhelming at first. If this is all you were getting all at one shot, that would be very overwhelming. But we've done these one at a time. And remember at the stomach, I said don't worry about the lipase at the stomach, so we can cross that one out. So for the saliva, this is for saliva, you've got salivary amylase. For the gastric enzymes, that's stomach. Gastric means stomach. You've got pepsin. And then we did the pancreatic enzymes. So these are probably the hardest to name. But the intestinal enzymes are pretty easy, aren't they? So make sure you know what juice each of these enzymes is in, and then what food group it acts on. You don't have to know details about how it acts on the food group, except for amylases break. It's not just carbohydrates, it's starches into disaccharides. Okay. All right. Now the last organ we're going to look at is the last place food goes, and that would be the large intestine. A lot of people interchange the word colon and large intestine, and it's okay with me if you do that too. The large intestine actually includes a little bit more than just the colon, but here you can see the different parts of it. Let's look at it on a picture. Right here is the ileum, so that's the last part of the small intestine, and it empties into the large intestine at this spot, which is another sphincter called the ileocecal valve or ileocecal sphincter. It's not at the very bottom or first part of the large intestine. It's just above it a little bit. So this little blind pouch that's below it right there is called the cecum. And extending from the cecum is some lymphatic tissue that you've all heard of called your appendix. Technically, it's the vermiform appendix. The word vermiform means worm-like, like a little worm there. But it's just lymphatic tissue, and if it gets infected and inflamed, it has a chance of rupturing, and if it ruptures, feces from the large intestine will get into that peritoneal space, which can be fatal. So that's why they don't ever fix it. They just remove it. You can live without that. You've got many other lymphatic areas of the body. Okay, but this first part of the large intestine is the cecum, that little blind pouch. All right, and then now you get to the colon, which the whole thing you can call colon is fine with me. But this is the ascending colon. And then as it goes crossways, that's the stomach is just above that, is the transverse colon. And then it takes a turn and goes down the left side of the abdomen. That's the descending colon. And then from there, it forms a curve. It doesn't just go up and down like this shows. It also goes back posteriorly. But that's the sigmoid colon. And that's what leads to the rectum and then from there to the anal canal. Okay. But all of it is considered large intestine. Okay. So, what does it? And then you notice one of the obvious things about it is it's bigger in diameter than the small intestine. Um, its musculature in its muscularis mucosa is a little bit reduced, especially the lengthwise muscle. And so you see it's almost the lengthwise muscle is almost like if you know what I'm talking about, if you sew, if you take a gathering thread and it gathers it up, that's what some of this is like. And it so it gathers it up in these little pouches. See these pouches? Those pouches are called haustra. That should be in your notes somewhere. Yep, the pouches. Okay. All right. This is significant. The mucosa has no villi, so it's smooth, like the stomach's mucosa is smooth. Okay. It has no villi. All right, but it is loaded with glands. They're called tubular mucus glands, and they have pretty much nothing but goblet cells in them. So guess what the secretion at the small intestine or large intestine is? Mucus. It's just a bunch of mucus. Okay, and so that aids in protection of it, but also especially the movement of the feces now through the large intestine. Um, while we're here on the anal canal, um, I want you to notice specifically this internal and external anal sphincters. The internal sphincter is smooth muscle. The external is skeletal. So the skeletal, of course, is what you would have conscious control over. Um, but that's a reflex initially when you're born, just like a urinary reflex. So a baby can't be potty trained until they have inhibitory signals going to that reflex center. We talked about that a little bit when way back in AMP1 when we talked about reflex. The defecation reflex is in the lower part of the spinal cord. And so if the rectum and anal canal gets filled, that stretching of the muscle in the wall is going to send an impulse to the reflex center, and it's going to come right back out to cause contraction of those muscles and the relaxation of the two sphincters. Okay. When a child gets to be about 2 or 3, their nervous system is developed enough to send inhibitory signals down to the reflex center to stop the continuation of that motor impulse. Okay. All right, here you can see the mucosa. And at first glance, it's kind of like that of the stomach. Today in lab, we'll look at a lot of this. We might do the micrographs next time in lab, but we're going to be comparing the stomach mucosa to the small intestine to the large intestine. They have some similarities, but they have some distinct differences. And one of the big differences with the large intestine are the massive goblet cells. Do you see all these goblet cells in there? Okay. Okay. But you don't see villi. Okay. All right, there you can see the internal and the external sphincter muscles. Seems like I've missed something here. Oh, what is the function of the large intestine? Do I even have that listed for you? Oh, I hadn't gotten to it yet. Okay. Okay. All right, so the functions, the biggest one of the biggest ones is water reabsorption. Okay. If you did not have a large intestine, then your feces would be very, very liquidy, like diarrhea. And over time, you're going to be dehydrated as a result. All right. When everything moves through the large intestine too quickly, that is diarrhea. And there's not enough time for that water reabsorption. So one of the dangers, the big danger of diarrhea, is dehydration. But then another is electrolyte loss, because you do absorb some electrolytes. You don't absorb any nutrients. There's no chemical digestion that occurs at the large intestine. There's no nutrient absorption, but you may have a little electrolyte absorption. Okay. You actually have a ton of bacteria in the large intestine. You've all heard of E. coli. Okay, there are some very bad strains of E. coli, but there's a really good strain in your large intestine, and it has a mutualistic relationship with you. You provide a nice, warm, and protected environment with lots of food for it, and then it acts on the feces It actually, they produce some vitamins for you, but they also cause, it sounds funny, putrefaction of the feces. And because of the bacterial action, those are the gases that we associate with feces. But they're very beneficial to you. So sometimes you hear about people taking probiotics. uh, That's, or the intestinal flora. That's these bacteria. Okay. Somebody who's taking antibiotics over a long period of time. Antibiotics, are designed to fight bacteria. They might, over a long period of time, they might be destroying these good bacteria too. So sometimes they suggest that you take vitamins in addition to those antibiotics to resupply some of the vitamins that you're not getting. Now, most of the vitamins you get from the foods you eat, but these bacteria can produce some. Hey, the movement through, you can relate to this, is more of a mass peristalsis. And then if you ever wondered what's in feces, a lot of different things. Tons of the bacteria, but they're resupplying themselves. Undigested materials, of those, a big part is cellulose. Cellulose is a polysaccharide found in the plant kingdom. It's not in animal cells. It's outside the cell membranes of plant cells. It's composed as what we call the cell wall. And for us, we can't chemically break it down. We don't have any enzymes to break it down, but it provides the fiber in our diet. So when you hear about fiber or bulk in the diet, it's cellulose. And it just passes straight through and aids with the movement. Okay, lots of mucus. Okay, let's look at this.
The presence of food in the stomach and chyme in the duodenum stimulates mass movements in the colon. Mass movements are integrated by the enteric plexus. They propel the contents of the colon toward the rectum. The presence of feces in the rectum stimulates parasympathetic and local reflexes that result in defecation.
All right, that's a lovely thought. Let's take a break now. Okay. All right. When we come back, we'll do the next chapter.